A muscle contraction, an action that we produce over and over each day in order to move our bodies. This can include everything from walking to lifting your pinky finger. But have you ever really wondered what takes place? In this video, we will discuss how a muscle contraction occurs at its most basic level. During a muscle contraction, this is what we see. But let's take a closer look. This model depicts a skeletal muscle. However, before we can really understand what is happening during a muscle contraction, we must understand the structures involved. Here we have an entire muscle which is surrounded by a thin layer of dense connective tissue called the epimysium. Within the muscle are found groups of muscle fibers called fascicles, as shown here. These fascicles are also surrounded by a layer of dense connective tissue called the perimysium. Within the fascicles, we find individual muscle cells, or myofibers, each of which has its own layer of dense connective tissue called an endomysium. The muscle cell, like any bodily cell, also has a cell membrane known as the sarcolemma. Each muscle fiber is made up of tiny myofibrils, which contain thick and thin filaments, as we see later on. And finally, these small blue objects are mitochondria, which produce ATP, the energy used in a contraction. Now that we have an understanding of the basic structural components that make up a muscle, let us take an even closer look. As the action potential or signal flows down the neuron, calcium channels are opened, allowing calcium to flow into the neuron. This increase in calcium then causes synaptic vesicles filled with acetylcholine to fuse with the membrane. Acetylcholine is then released from the synaptic knob into the synaptic cleft. When two acetylcholine are picked up by receptors found on the motor end plate of the muscle fiber, ion channels open, allowing sodium to flow in and potassium to flow out. This increase in ions initiates an action potential on the sarcolemma, which travels down and into the T-tubules. This stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum. Calcium then floods over the thin filaments, binding to troponin and pulling the tropomyosin off the active sites of the actin. The high-energy myosin heads of the thick filaments then leap to bind on the exposed active sites of the thin filaments, forming a cross bridge. The myosin heads pivot to the low-energy state, releasing ADP and moving the thick filaments over the thin filaments. This is known as the power stroke. ATP then binds to the myosin head, causing the head to break free from the active site of the actin. The myosin head then becomes energized as ATP is converted back to ADP. Shown here is the sarcomere, the basic contractile unit of a muscle fiber. It extends from Z-disc to Z-disc. As the thick and thin filaments slide over each other, they pull the Z-discs towards each other, thereby shortening the muscle fiber. Once a contraction is complete, calcium is pumped back into the sarcoplasmic reticulum and attaches to calsequestrin, which holds calcium until it is needed again. This allows tropomyosin once again to cover the active sites of the actin, and an enzyme known as acetylcholine esterase then removes acetylcholine from the receptors of the muscle fiber, and the muscle relaxes.